So the fifth key area of Unit 1 is learning about the structure of the genome. And it's quite a short key area, but it is important because it sets up the next couple of topics that we're going to do. So first thing we need to do is establish what this word means, the genome. And as it says, the genome of an organism is its entire hereditary information encoded in DNA. So when we see the word genome, we need to think, right, that's all of an organism's DNA, the whole thing. Then we've got this word here, genes, and it's important not to mix the word genes and DNA up because they're different. DNA is the whole molecule of hereditary information, whereas genes are just a part of the molecule of DNA. So genes are sequences of DNA bases that code for proteins. So if you have a look at this diagram down here, we've got the cell. Inside the cell, we've got the nucleus, and inside the nucleus, we have chromosomes. Chromosomes are made from DNA, which is here, it's unraveling. This is the DNA molecule, this double helix structure that we should be familiar with. And a gene is basically just a section of that DNA molecule. And as it says, it's a sequence of DNA bases that code for a protein. So basically, it's like a set of instructions to make a particular protein. So if the entire DNA molecule was a recipe book, a gene is just one recipe from that book. Okay, so it's just the instructions to make one protein, whereas the DNA molecule is the instructions for all the proteins. The genome is made up of coding and non-coding sequences. So we come across these before when we learned about transcription and translation, and we learned that between the two, they go through a process called splicing, in which we've got the exons are kept in, and the non-coding regions, which are called the introns, are removed. And then you splice together the exons so that you just have a chain of exons and there are no introns left, because you only need the coding regions of DNA to go on to the next stage. So these non-coding sequences, as it says, these non-coding sequences include those that regulate transcription and those that are transcribed to RNA but never translated, such as tRNA, rRNA, and RNA fragments. What does this all mean? So why do we even have non-coding sequences? Well, first of all, it's some of the reasons to that remains a mystery to scientists, something that still needs to be learned about and studied further. However, we do know some of the purposes of these non-coding sequences. So they can help regulate transcription, which is obviously very important for protein synthesis. And those that are transcribed to RNA but never translated, so these are types of RNA which go through transcription, but they don't go through the process of translation. So obviously this is referring back to previous topics we've looked at. So if any of these terms you're unsure about, just have a little look back at the last couple of key areas that we have learned about, and that should refresh your memory. And that's really key area 5. It's very, very short, but it does lead on to our next one, which is mutations, which we will do next week.